Hello, everybody. I'm Sai Lakshmi Subramanian. I'm the program manager of the Cancer Genomics Cloud. I'm very, very happy to welcome you all to the second webinar of the CGC monthly webinar series. So during last month's webinar, I gave a one-slide preview of how the CGC can be used as a data analysis platform for the PDXNet Data Coordination Center. And today, you're going to be hearing a deeper dive into the PDXNet story. So I'm delighted to introduce our expert speaker, Dr. Dennis A. Dean II, who is a principal investigator at Seven Bridges. Dr. Dean is a translational scientist who strives to bridge the gaps between theoretical and practical applications of patient treatments. His experience includes leading national initiatives, supervising large-scale data analysis projects, and developing novel methods and algorithms for real-world applications. He guides his team towards a collaborative strategic vision while also expertly handling operational details of his projects. Dr. Dean leads the Seven Bridges component of the PDXNet Data Coordination Center in collaboration with Dr. Jeffrey Chong at Jackson Labs. So thank you so much. I just have a few logistical items to share. As you can see in this slide, all the resources, including information about the upcoming webinars, slides, and recordings will be made available in the CGC website. So please feel free to bookmark this. Also, save the date and time. These monthly webinars happen on the same day, the fourth Wednesday of each month at 2 p.m. Eastern. Also, if you have any questions that you want to ask Dennis, please use the chat box for asking. At the end of the webinar, Dennis will be answering those for you. And at the end of the webinar, we'll post a link to a survey. So please fill that survey. It's very helpful for us to structure our webinars and also get your feedback. And tomorrow, you will be receiving a link in your email with a link to view the recording, and also the feedback survey link will be there as well. In case if you miss filling it today, you can do that tomorrow. With that, over to you, Dennis.
Okay, thank you, Sai. Hello, everyone. It's a pleasure to present our work with PDXNet. It really is an exciting project. We have maybe 20, 25 PIs that we work with. At last count, it was 130 people involved in PDXNet. And it's just really a giving set of researchers. And so I think it's a really good case study on how we bring people onto the CGC and what are the things we needed to do to make the platform most useful, or basically to advance the goals of the PDXNet community. So for today, I have basically a three-part agenda. What I'd like to do is give you some background, some motivation, and then I want to go through the PDXNet, the organization, what are the goals of PDXNet, and then I want to show you how we do resource generation, how do we collect data, how do we build our workflows, and I want to particularly focus on the innovative tools that we develop to advance PDXNet. And I'll end with the PDXNet portal, which is basically keeping a running tab of the resources as they are generated. And this is the first R-Shiny app that we're working on at Seven Bridges, so I thought it would be good to go in maybe a little more detail. Lastly, I want to go over a conceptual model of thinking about how we bring consortia onto the CGC. And the model is kind of a tool that I use to help me guide effort levels and really think through how are we doing in the resource generation pipeline. And the idea is relatively simple. You can't wait until the paper is published to generate resources. You kind of have to develop it as you're going. And so as I go through the talk, those are the types of things that I'd like to emphasize. So some motivation and background. So for this group, this is not news. Back in 2016, then Vice President Biden announced the Cancer Moonshot initiative. And the idea is to reduce the amount of time to bring solutions to cancer patients. And the idea was, can we squeeze in 10 years of research into 5 years? There were a lot of components, and the part that I'm going to talk about today is how do we make data available and usable as quickly as possible. And I think somewhere on this slide, it said something like 0 time to sharing. And so I want to tell you about our process doing that. Just a quick overview for background on the Cancer Genomics Cloud. The CGC is an NCI-funded cloud platform. And the idea here is that if you bring tools and data and people on the cloud, you can then collaborate more widely. One of the major advantages of the CGC are the data sets that are there. And we have over 3 petabytes of data. And just for reference, it would take a really long time to download that, like in the order of months. And so having computation and data on the platform is something that can accelerate research. So it's feasible to just jump on today and actually start generating results pretty quickly. One of the things you'll notice about all these data sets, for those familiar with the field, is these are really well-known data sets. Some of them have been around a while. So our team is really adept at how we take these large data sets and bring them onto the platform and share them. One of the challenges in the PDXNet community is that it's basically a relatively new research area. So there were no standards for how, or no well-used standards for how we share and collect these data. And so given that, I was tasked when I took over the PDXNet engagement, and this is the question that I really thought about a lot. How do we automate data processing from data collection to data publishing? And there were some challenges. So the data set isn't well defined yet. The other thing is we opted to not use our engineering teams. And so I had to reimagine what a team would look like that's US-based. That was a requirement of the graph. And over time, I broke down that question into 3 related questions. One, how do we decrease the time required to validate data? And I also want to add collect metadata, because that's really what's going to make it useful. The second was, how do we work towards developing functional standards that lets us move forward? Our original design often revolves around ontologies, and I quickly thought this was just going to be too complex to implement and change on the fly. So I had to really think about what is a functional standard. The third part is, and this was the PDXNet charge, it's the charge of the Cancer Moonshot. How do we get this data available quickly? And I do want to say we were able to share data as collected almost immediately on the CGC. The harder part was, how do we make it publicly available? We needed better metadata. We had to be submit through dbgap. And so that's a large part of the story that I want to talk about today. So what are the tools that we developed that help accelerate that process? So let me give you some overview of the PDXNet community. So let me just jump in right away. So I've already said that the PDX core Moonshot is a collaborative group aimed to develop preclinical testing of pharmacological agents. For those who don't know what PDXs are, they're patient-derived xenografts. And so basically what you do is you take a tumor sample, and I'm now talking about the figure in the lower right, and then you can embed it in a mouse, and you can embed multiple tumor samples, and you can also passage those samples. So you could take a sample from mouse A and then pass it to, it's not labeled, to the mouse in passage 1. Kind of like when you grow cell lines. So you can grow tumors over multiple passages. And our goals are very, very clear. We wanted to generate, help in the generation of 1,000 new PDXs with corresponding sequencing data. So the question wasn't just the models and the results from pharmacological treatment. It was also, can we use the sequencing data to gain greater insight into the pathology of cancer? We knew that we would have to develop new methods and that we would likely establish new models. PDXNet is a distributed group of individuals from coast to coast. And I have the PDTCs labeled here. So really working on the CGC was an advantage because everyone could log in and everyone could upload data, download data, and do analysis. And I'm very excited to say at least 4 of the centers out of 6 do regular uploads and downloads and analyses on the CGC now. I think it's important to give you a sense of the complexity, so I'm not going to go over the details of the schematic. I just would like you to understand some of the components. So on the left side of this figure, these are all the development and trial centers. I refer to them to PDTCs. In this talk, I'll likely just call them research groups. We have a well-established organization of committees that work together to generate a whole range of deliverables where the models and the sequencing data are really the primary outputs. We're also really lucky as a consortium to have a coordinating center because not all consortia have their own coordinating center. So we collaborate with Jackson Lab on the coordinating center, where Jax really focuses on the scientific component, and we really think about how we make resources available and generate a large data set that can be used for scientific research. We also collaborate very closely with the patient-derived model repository, that's the PDMR, who has a really large depository available on the web of sequencing, PDXNet sequencing data. So working with them is great because they give us really great templates for how to move forward. Lastly, our goals are to share this information, the models and the results, so that they lead to new clinical trials or letters of intent for clinical trials. We also have research supplements, and so that's the bottom part that was just added. We have PDXNet researchers work with 11 institutions to help evaluate more therapeutic options and to develop novel therapeutic concepts. I want to give you a sense of the research, even though I'm not going to dive deep into the research in this talk. So research, collaborative research is organized by pilot studies, and we have 3 years of pilots. On the left side, I'm trying to give you the sense of what these pilots are. So in 2018, the first pilots were really just about robustness. How good is a PDX model for cancer research? We then added a lot of pathway analysis related research in 2019 while continuing some of the robustness. And then lastly, just in the last few months, we added a whole new set of research that really revolves around new data types. So we've added proteomics, image processing, and there's a couple of machine learning related questions. But to be very clear, we really want to go towards mechanisms. We're just applying new ways of thinking about how we can answer questions in this domain. The last thing I want to just mention about the research is that whereas the coordinating center really led some of the earlier research in robustness, all research now is really coordinated, or a large portion of it, by the PIs developing working groups and communicating with each other. And if you ever want to see what it's like, if you join one of our PDXNet network-wide meetings, it's amazing to see the collaborations that come out of these discussions. So let me just give you 2 quick examples of research before I move on to resource generation. So this is the first study PDXNet published last year. And the question we had was, if you give someone models and you tell them test a drug, will they get the same response? So give no information about the experimental protocol, just say, here's the drug, here's the mouse you use, what do you think is going to happen? And what we found is that we could do multi-center trials across all the centers, and all the centers got the same results. So on the top line are the results from the PDMR. That's the established sharing mechanism through the NCI. And you can see we had 3 different types of models: no response, partial response, and responsive models. And you can see, if you look down to the second line, the results are pretty much exactly the same. Like, it may differ a little bit, but conceptually, you could then characterize studies the same. And so this was exciting. And building on this, a second study was defined. And the question in the literature or in the field is, are PDXs stable? Are they a good reflection of actual tumors? And we published just in February in Nature Genetics that the answer is yes. So using 1,451 PDX samples across 509 models, so a really large amount of data, and Jeannie Lee at JAX just did an amazing job on this. The important part to see is that what I'm showing you are correlations between patient tumor samples and PDXs for C and V, and it's pretty consistent across multiple passages. And so this is just foundational research, and I'm excited in the coming years we'll show you some of the great science that's also coming out of PDXNet. So let me talk a little bit about resource generation and publishing. And so this is something Seven Bridges does all the time. Even with doing it all the time, there were emerging challenges. So I'll tell you what was standard and what are the things that we had to kind of tweak to make work. So what data do we have? So we've collected about 55 terabytes of data on the CGC. A large portion of that is the PDMR data that was already existing. However, by co-localizing the PDXNet data and the PDMR data, we're able to do much larger studies right from the beginning. And we can also do preliminary studies or validation studies with the PDMR data. We just got a request to use the PDMR data yesterday, for example. So it's something that's used quite regularly. PDXNet has generated 273 new models across 6 contributors and 33 different cancer types, 2,800 new sequencing files. And to give you an example, that's about a third of what already existed in the PDMR and half of the whole exome sequencing file. So we expect PDXNet to really contribute to the publicly available PDXNet data set. Just some supporting information. We also compute a number of metrics and QC on all the samples. And although we didn't start with a standard, all our work now is supports the PDX-MI standard and the PDMR metadata standard. I'll talk about that a little bit later. I also wanted to say that we have a wide range of cancers, but we do have strong enrichment for breast cancer. And we are also adding imaging data and tumor volume data. So the workhorse of genomics data are the workflows. And so we developed part of the temozolomide manuscript, a set of standard processing workflows that were validated and compared against other published workflows. And so we now have 12 publicly available workflows that do everything from single samples to batch, whole exome, tumor normal, just tumor, just normal. And those are all available in the public portal on the CGC, which you can see on the left. An important thing to note, and this is kind of the CGC strategy, is that all our workflows are represented in common workflow language. Now, if you are not familiar, Common Workflow Language is a way that you can send workflows to other places to run. So it's reproducible and portable. The picture on the right side is a schematic of a workflow. It's also kind of nice to just get a sense of the complexity and the ins and outs of a workflow to look at it visually. This is really important because we're seeing that quite a number of people want to run these workflows, not just on the CGC, but locally. And that makes sense, right? Because there's clinical data you might not want to move to the cloud, or you may be prohibited to move to the cloud. So we really are seeing the need to have portable workflows. It's not just about the workflows. Our researchers asked for detailed documentation, and we developed them. So on the left, you can see the workflow description document. It must be 50 or 60 pages at now. And then we have a sequencing quality control protocol in the middle. Once again, another 40 pages of all the details of how we process the data. All the parameters are listed in the workflow document or can be pulled from the Common Workflow Language. So one of the great things about the CGC is not only can you run workflows through the visual interface, you can also run them through an API. So basically making command line calls. Some of your big people, this is the only way they work. And so what I'm showing on the right are the commands. Let me look. These are Python, I believe. Python commands for executing workflows remotely. And this can be used, well, I'm kind of a geek at heart. This is just exciting. But really what this does is it allows different types of users to access the CGC. So before I go through the tools that I believe are adding to our ability to process data and make it available, I want to give you an overview of our processes. So what I'm showing right now are the model development flow. And so basically on the left side, it's the PDTC, or the research group. They generate new models, and the long-term plan is to get it on the PDMR FTP site. That process generally takes about 2 years. And so we wanted a slightly faster model well, actually the PDMR offered or agreed to kind of accelerate sequencing. And we also envision improving the quality of the metadata. And so what you're seeing at number 1 is this PDMR model submission format. And this submission format actually is an R version, a Shiny app version of the paper form that the PDMR uses. And what this app allows us to do is pre-check for all the obvious mistakes that might be made in a submission. That information is then shared with a second model reviewer from JAX, who says, yeah, the science makes sense. Once those 2 checks are done, the model information goes to the PDMR, where they have their own review process that then leads to the model being archived at the PDMR, and then the metadata gets shipped to us. And if sequencing is done, it gets shipped to us pretty rapidly. And the important part here is this is allowing us to get in really high quality metadata, which was a challenge early on. And it was actually a request of our researchers. Can we have one flow? And so this is the flow that we put in place, I think, sometime this past summer. Now, the second part of this diagram is the sequencing component. And so I'm going to go over this one pretty quickly. But basically on the left side, we have the primary inputs, which is essentially sequencing data and metadata. We're also accepting new data types, the images and tumor volume data, and we have standard processing. The important part for this discussion is that we developed a dbgap file app, which allows us to process the samples directly from the PDXNet portal to develop forms for dbgap submission. And the idea here was to help accelerate the publishing part. And so if you see the A, B, and C, these are the primary outputs of our sequencing data generation. So we want to publish scripts and workflows, we want to catalog resources, and we want to make sure that we use the approved standard for sharing genomics data through dbgap. And so if you look on the right, this is more of a public view of where we're going towards. We want to have immediate access to workflows and scripts. We also want all of the metadata updated in real time on the PDX portal, which I'll show you later. And so this is a new shiny app again. And it's really important, not just for short-term storage, but where is the long-term storage going to be for this data? So really early on, we started discussing with the Cancer Data Service, who aims to make genomics data or cancer-related data that doesn't fit necessarily in a specific place, more widely available. So we started talking with them, it must be a year ago. And so when they went live back in January, we touched base with them about PDX portal, or about sharing PDX data, and that is actually in process right now. So the idea is that if the data is in the CDS, that's the bucket kind of all the way near C, it's then part of the NCI infrastructure and then can be shared more widely, not just on the CGC, but on the other two cloud pilots as well. So for the remaining part of this section of resource generation, I want to highlight the 4 novel apps that we developed, and I believe it really just facilitates data sharing. The first app is the PDMR model app. The second is the Globus uploader. The third is the dbgap file app. And lastly, I want to do an overview of the PDX portal metadata, so the PDX portal. Before I go on, I just want to say, it would be really great to do this interactively in the portal, but I opted to do screenshots so we could just go quickly. So I'm not going to go through all the details. I really just want you to get a sense of the flow of maybe how this would work, so that we can leave more time for questions at the end. So PDMR Information Worksheet. So the PDMR has its own paper form for submitting models to the NCI. And so my team developed this app that allows us to, if you click here, basically fill out the form. This is, just a electronic form of the form, which basically collects model information. And what's exciting about this form is all the metadata that you need to analyze and use the data is already well laid out. So by working with the PDMR, we're also ensuring that the PDX portal has really great metadata. And I just said this, but I want to say it again because I think it's really important. When I did my first question, I talked about automation. I should be very clear, I'm not a fan of automation. I'm a fan of people-assisted automation. So at each step, there is someone looking at the data, and I just wanted to state that again. So if you look at 1, 2, 3, we have an electronic form that does some checking, but then we have 2 people that check subsequently, one in JAX and one in the PDMR. And this was working relatively well considering that it's not completely automated. Yes, there are many things we could do to make it better, but we believe that we're accelerating the process of model submissions. So Globus access. So one of the first challenges that came up was the NCI said, we have this data, we want to share it with you, and they wanted to share it through Globus. And NCI, the CGC has many uploaders, but we don't have a Globus uploader. So I tasked a couple of people on my team. The first one was Ryan, and then Jeff on my team started working on this. And just to give you an idea of what the Globus uploader looks like, this is a screenshot. It's really a beautiful interface. It's really easy to use. We just don't support it. So Jeff came up with a really great solution. So he wrote some scripts, some shell scripts locally to get a sense. And then what he was able to do is actually run it from the CGC. And so now we have a really good mechanism for moving data from the NCI to the Cancer Genomics Cloud. But there's a second part that I think is possibly even more valuable. We now have really good instructions for doing this, and we see sharing these instructions is one win. So all the PDXNet researchers have access to this. We're now discussing how we make some of these scripts more publicly available. But really, what's great about this, we now have a really good mechanism for communicating with our engineers and the people building and extending the CGC day in, day out. So this is really a great opportunity to kind of prototype something that could be a feature later down the line in the CGC. So the dbgap submission form suite. If there are experimentalists on the line, they're familiar with the dbgap submission process. I skipped over the schematic. It's a multi-stage format, but one of the steps involves having a detailed list of the samples with associated metadata and funding information. And what you'll see in a second is that those sheets are actually quite complex. And what I hear from people who do this all the time, getting these sheets accurate could be a challenge. And so when we thought about what is going to accelerate data submission, we thought generating these, not forms, but these spreadsheets would be really important. So let me just walk you through that process. You basically open up the app. This is the user selection page. So it's basically what is the manifest of data that you want to use. And we already have those collected by PDTC. So we're able to interact with our PDX portal, pull the manifest, and then automatically review the information and the context. So these are contacts for researchers. And then generate this Excel spreadsheet. And this spreadsheet has 9 different tabs that has all the sample information, and they're correlated, right? Like they're linked by IDs. And so you can imagine doing this by hand could be quite time consuming. And my team is doing this in like under 30 seconds these days. So we're very excited about this, and this is another tool that we're thinking about how we integrate this into our platforms. So the last of the innovations that we developed is the PDX portal. It's actually the first. One of the exciting things about working on the PDX portal is that we could be iterative. So when we work with our more established teams, we really work on a longer term scheduling. There could be a build over 2 years. What we learned about working with our PDX researchers is that they really are iterative, they're agile, they want to know. And it's not that they want to know because it's fun. Every one of our researchers wants to find relief for their patients. And so the PDX portal is growing to be a way to access the exact status of what we're collecting and what will be made available. So this is the landing page for the PDX portal. On the right side, we have a few images so you can get an overview. The portal is organized into 3 sections: resources, analysis, and tools. And I'll just give you a quick snapshot of what these look like. So these are data resource examples. On the left is the sheet for the PDMR repository. And you can see we have overview stats at the top with donut plots underneath. So you can see we have 9,492 files. If you look, there are, is that 78 disease types? Wow, that's a lot. And then on the bottom, you can see by disease type, the whole exome and RNA-seq data types. I believe the blue are whole exome and the yellow are RNA-seq. On the right, this is brand new. It was put up last week. This is the image repository that is just growing. This is a direct response to the pilot committee that's looking at how we incorporate images into PDXNet analyses. I'm just going to give you one example of an analysis resource. This is homologous recombination deficiency. I can't say that 10 times fast, but I'll work on it. And what's nice about this page, and the other pages are very similar, is you could then filter on the left by data type, sample type, experimental strategy. And so I wanted to give you a sense of what is the type of information that you can interactively generate. So I was really curious on what is the diversity of samples. It's a question that comes up pretty regularly, and PDXNet is also funded in part with increasing the diversity of samples. So you can quickly generate this donut plot that I'm showing you, and the donut plot is showing the ancestry of the sample. So it's basically 93% European. Maybe you want to call that 7% diverse. And I was curious, if you looked at just the PDXNet samples, what would the diversity be? And when you look at that, you go, wow, this is great. So it's 60% European, 40% diverse. This potentially can have a great impact in the diversity of publicly available samples. And so you can quickly create the next donut plot that has all the data together. And you see, by adding the PDXNet data, we're going from 93% to about 79% European, or said inversely, from 7% diverse to about 22% diverse. And this is exciting, right? And so the exciting thing for me isn't the numbers. The exciting thing for me is then we know the status of the data set at any time, and this will allow researchers to plan studies, to plan grants, and also to plan recruitment. So if you know you want to study a particular cancer, you at least know what your starting point is. Before I move on, I just want to thank the Ancestry pilot. One of my roles, I believe, is to push forward. And I asked the Ancestry group to come up with a preliminary result by the new year. It was an aggressive timeline. They had just gotten started, and they did it. They created a workflow. They generated the results. This was done, I forget, Lewis from UC Davis is the PI leading this with JAX. And then Brian Sanderson from JAX really did a lot of the analysis. And then my team made this publicly available very, very, very quickly. And so I just wanted to thank them before moving forward. The last tool that I want to show you is an interactive analysis example. So this is a workflow calculator. By doing so many runs, we know about how much it costs to execute these workflows on the cloud. And it's probably our number one request. How much will it cost to analyze these sample? And so this calculator will do that automatically for you. You just have to put the workflow, and then in the middle, the number of samples. And then if you don't know the size, it will approximate the size for you. And then it'll give you a range of costs for storage and computation. So I want to end with how I think about supporting teams and groups on the CGC. And so I think about pillars of excellence. You know, if we want to have a great resource, I believe we have to get each of these steps right. So the first is platform excellence. Increasingly, we want to collaborate with large teams that aren't in the same place. So I see platforms as a way to do that. And there are a bunch of tools that platforms come with, like the CGC, data sharing, computation, and I added Cancer Data Service because it's really important to integrate into existing and emerging infrastructure. The next pillar is strategic efforts. Strategic efforts are the areas that you have to get right or you're not getting anywhere. And for PDXNet, it was really data excellence. How do we have great standards, great metadata, so that we can combine all those data sets and make them useful? Early on, it's hard to believe when we started, there were no standard workflows. And now we have standard workflows that we're using regularly. I'll just use those two examples. So translational excellence and operational excellence switch places often in this diagram, but I decided that you really got to get the translational questions right first. What are the ontologies and standards that we need to kind of merge with the community? How are we going to share this data? And then more importantly, what is the clinical translations we aim to see? Thinking through these questions early on then can influence how we do things operationally. I already showed you a lot of the operational work. We now have standard processing for data collection and processing and publishing. And then lastly, our goal is to publish data or to make data available, but at the end of the day, we really have to show scientific excellence. We need to be able to demonstrate that the work of PDXNet is valuable and influencing the community. So one of the really interesting things to me is as I was generating this diagram, where do we put analysis? And then what I realized is that we need analyses at each of these stages, or each of these pillars require analysis. The other thing that I realized is that these analysis steps is really the seeds of the resources. So if we can get this right, we can then think about how we generate resources, and not just the sequencing data, but there's an opportunity for sharing standards and scripts if we can just be a little more systematic. Like, to give you a quick example, so if we have a template, maybe we can use that to help design future features. So I want to end with the question I started with, and that was, how do we automate data processing from data collection to data publishing? And the answer is very clear to me now. We have to collaborate to establish small multifunction teams. I have a team of 2 and 3. Jax has a small team. But by being multifunction, we're able to collaborate and develop these tools and not only help with the science, but then help with the resource generation. The second is we just have to have a functional standard. I think early on we were trying to get the best standard, get an ontology. I think we just have to get started, which is a little troubling sometimes. But I think it's the way we have to do and move towards an established standard. And so I'm happy we have a more established standard. But for something like histology, we don't have a standard yet, but we're still able to make data available and share it. Has to be gold. So functional is really important here. And lastly, I believe we have to collaborate to build tools while the science is being done. I think waiting till the end is just problematic. It has to be, we have to show commitment to advancing the entire field right from the start. So just to summarize and to conclude, the PDXNet is well on its way for generating the resources as anticipated in the grant. We can share data very quickly internally. I believe we want it to be faster, but we now have a system in place that will allow for sharing relatively quickly. So the bottleneck will soon be just doing the dbgap application, and Sarah on my team is really helping with that. I'm really excited that we are increasing the diversity of publicly available samples. So once we're able to get all our dbgap, our data processed, we're going to be very happy and excited to see this data used. So, we did generate a semi-automated data collection workflow. The part that I really want to emphasize in the conclusion is this really stems from collaborating with many of the existing infrastructures in the NCI. So we work very closely with the patient-derived model repository. We look through all the dbgap processes and try to figure out how we can best interface. And then lastly, I have to thank the Cancer Data Search for really walking us through what it was going to be like to submit data even before all the details were known. So we're able to plan for the submission. And I just want to end with what I believe are the most important things to remember when we think about accelerating data access. We have to collaborate to establish small multifunctional teams who can be agile, because there are going to be issues, there are going to be unexpected events, and we need teams right there to help push things along. We need standards, we need to share them, and we need tools that help enforce them. And then lastly, we need culturally to see science and data resources as being equivalent. And something that we provide equal time to. At the very least, we need to do it at the same time. So with that, I just want to say thank you. I want to acknowledge the PDXNet Consortium. EuroPDX collaborated in some of the early publications. They're great collaborators. The JAX team on the right and the Seven Bridges team does all the work. I have to say, I don't do this work anymore. Jeff, Sonar, and Sarah on the right really just make things happen. It's just so amazing to see their ideas just come to life. I have to thank the public programs team, especially the CGC program managers, who really are there to help all of the way. Our funding is there. And lastly, I just want to thank all the PDX researchers who are just aspiring to work with, and particularly Jeff Chung, who you could see kneeling kind of in the front on the right, who's the JAX or one of the JAX PI. There's times where we speak almost every single day. And so he's just been a great collaborator to advance this program. So with that, I will take questions. Thank you.
Thank you so much, Dennis. That was an incredible presentation. I loved hearing the story of how PDXNet evolved and how the Data Coordination Center has been successfully using the CGC for all the analysis. And more importantly, one of the main takeaways for me was how beautifully you've come up with best practices. When standards were not there, you've come up with new standards, and also all the documentation, new tools and workflows that have come out of this this collaborative effort. I think that's an incredible takeaway for me. And also seeing your latest Nature Genetics paper, it gives more scope for researchers to take on this research, right? Like in this PDXNet area, right? PDX area. So with that, I really express my heartfelt thanks to you for sharing this story and also this wonderful presentation. There's a feedback form that has been posted in the chat, so please provide your feedback on this webinar. We have a couple of questions, so I'll read out the questions. One of our attendees has asked, how can we access data today? Any plans to add other data sets such as small RNA-seq?
Yes, so we're finalizing the dbgap submission process. And so we have 2 studies registered. I think at least one of them is live. The second one will be live soon. We use just a small number of samples to establish the process, but we're now adding samples. So we are expecting, we have 2,800 sequencing files. We expect hundreds of those to be, if not like 1,000, in the coming weeks to months, pretty quickly. And then it's really down to the dbgap process. So what we can do is we know we'll have a question in the survey if you want to be contacted when the PDXNet data goes live. The other thing that you can do is you can reach out to our researchers. We're always collaborating with PDX researchers. So as long as there's some synergies, there may be ways to collaborate with the data. And then the second part of the question, so yes, we're constantly thinking about other types of data sets. We're thinking about proteomics. There will be some proteomics collection. There are images. We have not, I think you said, was it small RNA?
Yeah.
We're not planning for small RNA-seq, but that doesn't mean that that might not change. The other thing that you might want to do, I can see the person's name, but I won't use it. The other thing you might want to do is PDXNet has supplements every year. They come out in January. So if you wanted to do a complementary study with the PDX researcher, I would highly encourage you to propose it and reach out. The researchers just love to collaborate, and this is clearly an important area. So reach out. And if you send an email, if you would like an introduction to either Sai or me, or respond in the survey, I'm happy to help make connections.
Excellent. Thank you so much, Dennis. The next question we have is, we are running a PDX project in Argentina. The whole exome data and metadata were uploaded to the CGC. How can we access the recommended PDXNet workflows and files required for QC and variant calling?
So yes, so all of those workflows are in the CGC public apps gallery. We can send you a link, but if you look for the public apps gallery, you can find it. And then you just search for PDX, and all the workflows are there. If you go to our PDXNet portal or to our PDXNet webpage, we have a description and then links directly. So if you find any of those three, you can get that. And I believe on the portal, we now have our documentation, which are detailed descriptions. I do have to say, Jess, the public apps gallery is quite detailed. And like everything, if you send us an email, we're happy to help you figure out how to bring those online.
Great. And additionally, I think we can post some of those links in the webinar resources as well, so you'll have it handy. If anyone else has other questions, please feel free to post them in the chat. We'll be happy to take them. So Dennis, while we wait, I had a question. So are PDXNet PIs and researchers interested? You mentioned they're interested in collaborative projects. So what kind of projects, like, can you give us some guidance so that people can think about these are, oh, maybe this could fit well with my research. I'll be happy to approach them. So what kind of collaborative projects?
Yeah, so the one thing, the commonality of all research projects are a clinical focus. So understanding combinatorial treatments, 2 and 3 treatments, addressing known challenges. So we know for certain treatments, there's only a 30% response rate. So those are the problems that PDX and researchers tend to go after. There tends to be a combination of an experimental component and an analysis component, so generating new research. Increasingly using large data sets like the PDMR, TCGA in collaboration with the PDX data are really emerging. So we're starting to see machine learning driven or pathway analyses, just more complex analyses. And I'll give you one more example because I think this is a very common approach. Understanding a particular pathway across cancers is also really, really common. And then using both whole exome and RNA-seq. And we're expecting proteomics to be added to that shortly. So that's just a starter. I think for most of the researchers, if there's a compelling need, they tend to try to find a way to collaborate.
Got it. Yeah, these are really exciting areas, machine learning, imaging, proteomics. I think there are incredible opportunities available to collaborate. Okay, great. So I don't see any more questions. So I would like to extend my thanks again to Dennis and also to all of our attendees for joining us today. I would just like to highlight a couple of things. We do have collaborative project programs as part of our CGC platform. So NCI has generously awarded grant cloud credits. So for up to $10,000. So if you're interested in reaching out for a collaborative project, so please feel free to contact the team. You can email us at cgc@sevenbridges.com. We highly encourage applications from students and postdocs, and we want to support their research. And lastly, if you have any help, if you need any help in learning things about the CGC, so there's the Knowledge Center. You can contact CGC support or also join us for office hours, which happen every week at 8 a.m. and 2 p.m. on Thursdays. You can go to the CGC website and there's a help option in the top menu, and you'll find links to different tutorials, user guides, and also the office hours. And finally, I would like to introduce the speaker for our next webinar session that's going to happen in March. We're going to have Dr. Zeros from the National Cancer Institute, as Dr. Xavier will talk about novel roles of microRNAs in tumors using the CGC. So please join us for the next month's webinar. It's going to be on March 24th. Be on the lookout for the email announcement and also webinar stage. With that, I would like to thank everyone for joining us today.
Thank you.